Thank you. I wanted to open this up with a brief anecdote from why this is really exciting for me. So the DREAM challenges actually started when I was in grad school, and my thesis ended up being a competition with my soon-to-be postdoc mentor in gene regulatory networks reconstruction algorithms. And then I ended up joining Sage for my postdoc briefly, and that's when Sage took over running the DREAM challenges. And I started hearing about how difficult it is to set up a challenge, which I didn't appreciate before as a kind of participant. And so today I get to talk about a challenge, and I feel like I've come full circle in that endeavor. So this is special to me. Without further ado, I'm speaking today on behalf of a distributed team between Sage Bionetworks, OIHSU, which is where I am from, and University of Heidelberg. And yeah, I'll jump into it. It's manually advancing. Do we have a pointer at all? It's not working. Yeah, I'm not sure.
That's Seth.
Seth to the rescue.
Let me. Yeah, there we go. Close those ones. Close those ones entirely. Yeah.
I am. This one.
There you go.
Perfect.
We get to your laser pointer.
Oh yeah, laser pointer.
There you go.
Awesome.
Your head.
All right. Okay, so in this particular challenge, we are focusing on two main components, the transcriptional and the epigenomic. And I think this is an audience that I don't need to convince at all, but in order to study complex biological systems, we want to understand transcriptional signal, and the epigenomic programs offer insight into the regulatory programs behind that as well. And so we've been discussing in this meeting about how prevalent the single cell technologies are. And so we now have the technologies to quantify transcript and single cell resolution, and also assess accessibility of chromatin at the single cell resolution. But these technologies come with challenges, and these challenges are unique, specifically due to sparsity because of the low amount of mRNA in cells that we are trying to sequence, and low amount of copy number for ATAC-seq. And so in an average experiment for single-cell RNA-seq, between 10 and 45% of the genes per cell are captured and detected. And for ATAC-seq, that goes down to 1 to 10% of peaks per cell that we can detect. The technical challenges here are how to navigate a PowerPoint. I now have a pointer, but I can't control my slides. All right. So we designed this challenge to explore the weaknesses and strengths of methods that are becoming more and more prevalent in our pretty much first step in our analysis. And so as we become more reliant on them, we wanted to understand not only how much signal we can extract and what we're focusing on, and I'll explain that later, but also what are the downstream effects of some of these methods. All right, so as we all know, this is a prevalent technology, and as the technology has been improving and maturing, the cost has decreased. We're now able to sequence and detect a lot more cells than before. And as these technologies have become more mainstream, it's easily become adoptable by research labs and industry. And so that leads to explosion in number of publications as well. But I guess most of us always faced the classic question of sequencing budget. On one hand, you have you could do, given that you can only sequence a fixed number of reads, you could do deep sequencing for a number of cells, and then you can quantify them really accurately, as shown in the diagram on the left. Or you can do, but then you're missing out on the population, right? Or you can do shallow sequencing and shallow sampling of the population, where you can capture some of the diversity, but that introduces a lot of noise in the data. And so as we make these decisions, they of course have implications on the methods that we use, and vice versa. Basically, different methods rely on different on richness of different types of data, and it's important to consider that. And furthermore, as we thought about this question, it really came about when I entered my new lab, and I asked a couple of my colleagues as I was starting to think about single-cell RNA-seq. I said, what pipeline do you use and what methods do you use to pre-process your data? And within the same group, there were about 5 people that were doing different things, and they didn't talk to each other. They didn't care about each other's work because what was working for them was working for them. And that didn't sit right with me. I just really wanted to understand why and what are the differences, and are we really looking at the same biology when we are looking at it from the lens of these different methods? So as we think about as I thought about evaluating these, I certainly wanted to do it in a reproducible, transparent fashion, which Dream Challenges provide. And I do want to see that these methods that we're using produce results that are consistent. And another thing that I think I didn't appreciate was really coming up with an unbiased evaluation criteria. You'll see hopefully that this is really difficult. And so I wanted to show this example from a recent review paper where they looked at the effects of single-cell RNA-seq processing methods after the data was processed. You could see all but this example here, where this is the raw data. What you're seeing here are definitely unwanted spatial effects that you can see also that correlate with the library size factor. And it's a simple test to do. These are just, you're plotting principal component analysis of the post-processed data. But I guess the methods were very focused on improving a particular aspect and never went to do something like this and see that they're actually kind of distorting data. So at the time that we started thinking about this, there were already a number of papers. I believe that when we defined this as a challenge, there were about 40 papers, and some of them were coming back from there's just such a range of technologies that these papers were trying to address, and these methods. And sometimes people would use they assumed inherent problems in data that no longer existed in the newer technologies. So there's a distinct difference between the different sequencing technologies that wasn't really being taken into account in how these methods are being applied. And so there are to date a couple of reviews that look at the one I'm highlighting here has they profiled 18 methods, and a more recent one from last summer profiled 8, but they partially overlap, and they profiled them on very different tasks, which is, again, kind of an endless quest of how you probe these methods as well. But I wanted to offer that kind of high-level summary of the types of methods that we are seeing as classified in this review. And so they talk about 3 general classes: model-based, where you actually have a model that defines your predicted behavior in the data, and you analyze the differences between that and the actual data. Smoothing-based methods that use underlying structures like graphs, and then data deconstruction, which could be these linear types of approaches, or deep learning methods that capture nonlinear relationships. And so as we thought about setting up this challenge, which shows 3 baseline methods that represented each of these classes. And they gave us a nice range of performance in our preliminary results and also space for improvement, which we were excited about. Okay. And so we went ahead to define this, the main question here. And that also evolved because in the beginning we were very focused on so-called dropouts, which I will also define later on. But what we realized is that the more general way to pose this question is really how do we pre-process our data and how do we correct the signal? And as I mentioned before, the question that's in the green box is really what was the scoring question for the challenge. But from the beginning, we had the intention to investigate what are the effects of these methods on downstream analysis. And that included really distinguishing what I will define again shortly between technical dropouts and true biologically zero transcript genes, as well as whether or not we are able to consistently identify differentially expressed genes and find populations of cells that behave similarly, to put it more generally, which people think about cell types, but more generally, really, some preserved behavior between groups of cells in our data. And so for sub-challenge 1, which is really the RNA-seq part, we went ahead and generated 3 data sets, and these are, hopefully I'll convince you that these were sequenced at really deep, at high depths, which is what allowed us to set up the challenge in the way that we did by downsampling these data sets. And so in the first experiment, we have 4 different libraries, which is why we will be kind of testing these as 4 different data sets, and we've labeled them DS1, A through D. And data set 2 and 3 were multiplexed. So what we went ahead and did is to downsample each of these, we call them 4 conditions, or you'll see them as 4 different data sets for data set 1, to these 3 different ratios of reads. And we did similarly for cells. Data set 2, we downsampled by cells to 25 in the respective 12.5 and 6% of the cells. And because of the depth of our data, these often emulate basically what an average experiment would give you. So we could really afford that. For data set 3, we did downsampling both by reads and cells. And for metrics, we decided to choose MRMSE as our main metric. In concordance, we always calculated Spearman correlation. We also had other metrics that we explored that showed very strange behavior, like geometric means. And it's interesting because we had to commit to a metric from the beginning, and you have to have a winner. But we're going to show you performance using both of these, and they don't actually agree. So for baseline methods, as I mentioned, we have these 3 methods that are representative of the 3 classes that were established in the literature. And I should mention that for data set 3 in exploring differential gene expression, we actually also generated on biological replicates of the same single cell data, bulk RNA-seq that we'll be using to validate these predictions. Similar to what Solly mentioned, this was a model to data approach where our participants didn't see the test data either. And so they were scored on data that was held behind a wall. And the reason for that was that we wanted to avoid overfitting and any type of really custom fitting to the particular data set that we were giving them. And all of this data, some of it has already been made available on the ATAC-seq, and the RNA-seq data will also be made available. And we hope that it will be of use to the community for further benchmarking. We impose some technical parameters for this challenge. This is more interesting in the second sub-challenge, which I'm not going to talk about today, because some of these methods are really computationally intensive, hence my curiosity about sustainability for the ongoing leaderboard earlier. For the RNA-seq, yeah, you can see these requirements, and similar to the previous challenge, there were some methods that met those, but they, in the later collaborative phase, when we reached out to them, actually explained that they had to undertrain their models because of the time constraints that we imposed. And in the collaborative phase, we removed these restrictions and allowed them to train their models as much as they wanted. And they actually showed significant improvements, which we incorporated retroactively into our results. Okay, so in this first plot, I'm going to be showing you results for all of these data sets that I mentioned before. If you remember, there were basically 6 that we downsampled, or 5 that we downsampled by reads, and 2 that we downsampled by cells. And so we'll be reporting the main metric here, NRMSE, small values are good. We will also be reporting concordantly Spearman correlation, high values are good here. So we're looking, the best performers would be on the left top corner, if there are any. And we're showing you downsampling to 10,000, 20,000, and 50,000 reads, respectively, here. Well, I did not see that coming. All the dots are missing.
Did you see them on your screen?
No, they're not on my screen. So this is a PDF plot that is not rendering. That defeats the point. Any wisdom?
So let me just. Huh. I'll be right back. I'll see if I can get another copy if that does happen. Sorry.
Well, I can talk about it. And you just have to believe me, I guess. What's your favorite method? Right. So what we saw was that there was pretty good consistency. There was basically, well, you can see the legend, and the legend is the methods are appearing in rankings. Starting here, this is a method that was outperforming Go Lab, followed by GLS5, and then you see the anonymous team 1. The mystery is that this is a team that submitted to the public phase, but that did not submit to the final phase, and they kind of didn't really stay in touch with us, even though we invited them to the collaborative phase. So we can show their results, but we can't name them, and they actually did pretty well. So you can see the baseline methods in this order. One of the baseline methods is almost at the end. I mean, they're kind of toward the end. Actually, I'm not showing scImpute here. The reason I'm not showing scImpute is because it didn't meet the constraints that we imposed. In the final phase, it required a lot more time than what we were allowing other methods. So we ended up removing it because it wasn't a fair comparison. Because a winner or a top performer has to beat the baseline method. And nobody could, but also the baseline method was requiring more resources than the others were allowed. So we removed it in this original consideration, but again, retroactively, we brought it back, and I'd show you the result, but you can't see it anyway. The point is, though, that all 3 baseline methods were toward the bottom of the performers, and so the methods that participated in this challenge actually did much better. Hopefully, the other plots will show. Yeah, that was the point, that scImpute is still beat by 3 methods, and then the other 2 baseline are toward the end. And scImpute, I believe, was the model-based method. Magic is a smoothing method, and Baselight, or the deep impute, is obviously a deep learning method. In evaluating the top performers, Sage and we are always concerned with whether or not these are actually significant improvements over any of the methods that are trailing close behind. So they actually do a fantastic analysis to compare base factors, the base factor of models that are near each other in performance. And in this case, it's not as exciting because nobody really came close to beating the winner. And if we look just at NRMSE, this is a very compelling winner. But this question, it all goes to how difficult this is, really, because when we look at Spearman correlation, we actually have two methods that do quite a bit better. And so in the downstream analysis of these results, we're trying to understand what are the benefits that each of these are introducing to the table, and what can we learn from this type of dichotomous behavior. One of the interesting answers here is that this ensemble model analysis that we did, which is essentially your methods are ranked in performance, starting from the best one to the worst. And we would integrate, you're starting with the first performer, which reads as 0 plus Go lab. And then in the second step, you integrate the second best performing method. And in this case, because we're looking at NRMSE, and reads, we were able to do an arithmetic mean of the results from both methods. And that's the result that we were putting here. So as you can see, for a change, this solid line shows the increased performance based on NRMSE. And after including the third method, that performance no longer improves. But what that tells us is that these three methods have some sort of complementary information that we can learn from. And we're excited to explore that as well. I wouldn't be too concerned about the behavior of the Spearman correlation. I realized that the methods here are ranked by NRMSE and not by Spearman correlation. So this really is not as relevant as I was thinking. But yeah, I think if we rank this by Spearman correlation and we do the ensemble analysis that way, I think that could be an interesting idea as well. I mentioned earlier dropouts several times, and I want to go into a little bit more detail now about this. So when you look at your single cell RNA-seq data and you see 0 transcripts, it can be due to technical issues, such as just really low amount of mRNA, like I mentioned before, that's really not captured in the final step of the technological process and sequencing, or it could be due to biology. And so what we define as a dropout here is the first case where you're observing 0 transcript, but it's really due to technical issues. And so in that spirit, we set out this ground truth to after correction table, where the ground truth, we call this 0 transcript that we observed in our full data set, which is very deeply sequenced, as you remember. And then if a transcript is detected, this is a non-0 transcript. And after correction, we look at whether or not the method produced a 0 transcript or a non-0 transcript, which corresponds to true positives here. It's a little bit unintuitive because we have 2 zeros. It's kind of like 2 negatives make a positive. So I like to think of them as the true positives are a success of maintaining zeros where they should, and the false positives are a success at maintaining, or this is an error rather, in correcting zeros where there shouldn't be. Oh no. Hey, this plot is showing. This is great. All right, so in the first plot here, we're looking at the false positive. So this is the error in correcting zeros. And as you can see, it's actually, it's okay. I think I passed that. Yeah, I don't think I'm going to have time to go back. I'm so sorry. Yeah, it's okay. So in this plot, you can see that most methods do pretty well, and there are a couple that have some sort of false positive signal here at the end. But on a relative scale, when we look at the true positives, those are also the same methods. The methods are ordered in the exact same way that you can see here. And those are also the methods that have actually pretty superior detection of the true positives or preservation. And so when you calculate an F1 score in that context, they actually do pretty well. And what's also interesting is that these two methods are actually doing they're achieving the exact, almost the exact same performance in this question of characterizing dropouts. Going further and thinking about clustering and cell types and this behavior of cells, really, like I said, more generally, if there are conserved relationships, or rather, if there are relationships between cells and groups of cells, are these still conserved after the correction methods are applied? Again, many metrics of evaluating this. We did a few, and they show pretty strong concordance, but I am choosing to show you the graph overlap. So what we did here is that for each method, we calculated a lower dimensional representation by principal component analysis, and we use that to construct a KNN graph. And then we look at basically how well the ground truth graph overlaps with the graph that's inferred from the corrected data. And we summarize that by Jacquard index, where high values indicate higher overlap. So you want to see higher values. High is good. Well, this plot is showing. That's really random. It's the same plot, but it's showing. That's exciting. So this is just a schematic of the experiments that we did. As you remember, you can see the data sets on the x-axis and the experiments that we did on the y-axis. Highlighted in yellow are dataset 1 downsampling by reads. Now highlighted in yellow is dataset 2, which we downsampled by cells only. And then finally dataset 3, where we were able to downsample by both. And to remind you, what you're looking at here in terms of performance is this Jacquard index of the overlap of the graphs that were learned. So high value is good. And on a relative scale, you can't really see much in this view for dataset 2 and 3. But there's some variance in dataset 1, so we're going to dive into that a little bit. This is showing essentially the same experiment. Again, on the y-axis, you have downsampled to 10, 20, and 50K respectively. And on the y-axis, the columns really represent the different data sets that we downsampled. This is data set 1. And what hopefully you can see is that if we just look here at 10,000 reads for this data set A, the methods are ordered in kind of increasing performance. We want this value to be as close to the right as possible. But there's also more noise here. What's interesting is that as we add data, overall, all of these methods are able to learn and perform better. And that's really, I guess, my message here. And that's consistent across different, these data sets actually correspond to different perturbations that we will describe in our paper. Oh, yeah. A point here that we also looked into other measurements of classic unsupervised clustering, and those are overall consistent on all the different evaluations that we did. One interesting example for dataset 2 was to look at, this is now showing ARI metric, which is how consistent your clusters were. So we're no longer looking at the graph overlap. And this data set is particularly biologically relevant because the perturbations that were done here result in very distinct graphs or clusters in the ground truth. So we just have more confidence in interpreting these and the consistency between the clusters here. And that shows an interesting performance where this method, anonymous method 3, wasn't really scoring that well up until now. So there are contexts where methods really perform very diversely. Again, this is another method. This is the baseline method that was almost at the bottom in other metrics, and LG Explore, which was second in the official leaderboard. What that goes to say is that the question is complex, the data is complex, and I think it's really hard to draw a conclusion. And also, we can set metrics for how well the data is similar or what we define to be correct. And then looking at downstream biological validation, there's so much interpretation to be done. Here's one of my favorite results, I guess, or it shouldn't be, but what we're looking at here is a classic volcano plot that we generated for dataset 1, where I mentioned we had 4 perturbations. These are perturbations of treatment and control at 24 hours and 72 hours. And, consider using one of these methods and not the other. Your paper will look very different, right? The significance here is, very variable. I think I'm kind of running over time with all the technical issues, so I'm going to wrap this up and say that the final ranking, I'm showing you the official leaderboard at the top. And my point here is just to show that changed quite a bit when we went back to kind of engage the community, because our goal was to really learn, and we just wanted to use every opportunity that we could and kind of engage people to participate. And like I said, we moved runtime restrictions to enable some of them to run. That showed much better results. You can see that the ranking is quite different. Some stats, and this goes to something that came up earlier in Soli's challenge. We actually had a phenomenal interest in the beginning. We had more than, I want to say I saw the number 300 something participants that were pre-registered. There was a lot of interest for our challenge. And I think in retrospect, when we required containers, we required all submissions to be submitted as Dockerized containers. That was, it's just a bit of a step still in the community to entering these. The other thing we didn't do is to release the testing data. And I think that was a deterrent. That's my guess. I'm hypothesizing here. But I think, because we advised people and the participants to use and utilize public data, but that's not as, I think, doing more work for them engages people, and it makes it easier for them to participate. And we also had the ambitious goal of actually, I talked about the RNA-seq part today, and I didn't discuss the ATAC-seq, which was an extreme challenge for us as well. But a part that didn't even make it to the public site was a multi-ohm challenge, where we had a multi-ohm data set for both ATAC and RNA-seq, which was, in my humble opinion, the coolest part of this challenge. So there were a lot of different steps that didn't end up working out the way we hoped. But in terms of lessons, I would say that's kind of what I learned, and that I would try to do better next time, because you can see the numbers decreasing. This is like exponential. At the end of the day, for this part of the challenge, we estimate that we scored about 1,400 models, and the ATAC part was exceptionally time-consuming and resource expensive to run. I'll mention the team. This has been a fantastic collaboration. I can't say enough thank you to this person. This is Rong Chai, and who is a software engineer at Sage. He's incredible and has done so much work. And this is Nick Calistri from Laura's lab at OHSU. They've just been, and of course, Jake. Everybody has been fantastic to work with. I'd like to thank my collaborators, everybody who helped us along the way, and I'm happy to answer any questions and show you plots later.
Questions for Olga?
Great work. Thank you. So I was wondering, was there anything specific about these data sets 1, 2, and 3 that made you think that one is good for downsampling the reads, and then one for number of cells, and then one for both?
Yeah, more we thought about, first we looked at the depth of the data and how many reads we got in each of them and how many cells, but also the experimental conditions, because we could use them to validate questions like clustering, like in dataset 2, where the rationale, I would say, was biologically driven. Yeah, and I didn't describe what the conditions are. This was a part of the challenge that we didn't disclose what the data was. It's data that you can then kind of reverse engineer because there's drug perturbations, ligand perturbations in this data, and time series.
Okay. Yeah, actually, you answered my second question.
Oh, good.
Too, which is like, what are the biological differences? Because some
Yeah.
data sets are more challenging.
Absolutely, absolutely. And the reason that we are so excited in downstream analysis is because we can now go ahead and ask these questions in these contexts where we have kind of this strong biological indication of how a method should behave and what the biology is. Yeah, so
thank you.
Yeah, it'll be in the paper.
Any other question for Olga? I think this is a very good example of a very difficult task, but that it's needed. We have all these scRNA-seq methods. I think a couple of days ago we saw there's this scRNA-seq tools page that has 1,500 methods, right? I think this is I've heard about this challenge and how difficult it has been. It has taken a long time. But I see the results, and I think in that sense, it is a successful challenge, right? We have a benchmark. I think it's going to be useful for the community. And this is what we try to do, right? We don't always overcome baseline methods, but I think all the work that's behind this and really trying to get an answer to a difficult biological question is important. I think is a step forward for the field. So thanks a lot for this, Olga, and for
thank you.
Going full circle.
Yeah. Different types of fun at each level. But I did want to mention, I'm not sure how many of you attended the rest of this meeting, but there was actually quite a bit of, there were several presentations on synthetic data generating tools. And notably in the review from 2023, there's a comparison between real data and synthetic data performance, and the methods excelled in synthetic data and did really poorly in real data. So I think there's still space for improvement.